Students, today we are going to discuss about the photosynthesis in higher plants and some of the important questions related to this topic. So as we all know that photosynthesis is one of the vital function, or we can say the unique function performed by the green plants. So here why I am mentioning it as green plants means there are some plants which are non-green and they are parasitic in their nature. That is, they depend upon others for nutrition. Whereas the green plants, they can perform photosynthesis and prepare their own food materials by using a cell organelle called as a chloroplast. That is a vital cell organelle present in the plant cells called as chloroplast, and that cell organelle contains a pigment called as a chlorophyll pigment. Apart from the chlorophyll pigment, there are some accessory pigments are there. Those are called as carotenoids. So in which carotenes and xanthophylls are present. So the chlorophyll, carotenes and xanthophylls, all of them are playing important role in the trapping of solar energy, and chlorophyll A is helping in converting that solar energy into chemical energy form, that is ATP. So here in the photosynthetic process, two steps are there. The first step is called as light reaction, in which the solar energy is trapped and converted into chemical energy form, in which two energy forms are produced. One is ATP and other is called as NADPH2. So these two are combinedly called as assimilatory powers. These two energy forms are used in the next step where the carbon dioxide fixation is taking place. That is the actual process where the carbon dioxide is converted into, or carbon dioxide is reduced into glucose molecule or the photosynthates. Okay, the first step of photosynthesis, that is in the light reaction, where the water molecule is splitting to produce oxygen. That is, the water molecule is undergoing oxidation to produce oxygen, and electrons are also being released. Those electrons are carried through various electron carriers and helping in production of the chemical energy that is called as NADPH2 and ATP forms. These two are utilized in the second step where the carbon dioxide is reduced to form the photosynthates, that is organic compounds. So this is a way how the photosynthesis is taking place in the plants where the green parts are present. That is, the presence of chlorophyll is playing a vital role in the process of photosynthesis. So this is a brief characterization that can be seen in the plants where photosynthesis is performed. So students, let us discuss some of the important questions. Now let us come to question number 1. The immediate product of carboxylation in C3 plants. The options are A. RUBP, B. PGA, C. OAA, D. GAP. The question is related to the C3 plants. So what are the C3 plants? So C3 plants are those where wherever the carbon dioxide fixation is taking place, the first stable compound that is formed is an 3-carbon compound. So that's the reason why we call those plants as C3 plants. And in this process, what happens is carbon dioxide is accepted by the primary acceptor, that is RUBP, ribulose 1,5-bisphosphate. So this will be taking up this one, that is this is a 5-carbon compound, and carbon dioxide, as we know, it contains only 1 carbon. So these are converted into the first stable compound called as PGA, that is phosphoglyceric acid. So here this is called as carboxylation step. Why we are calling it as a carboxylation step means because the RUBP is taking up carbon in the form of carbon dioxide. Taking off carbon is called as carboxylation step. So this is the first step of C3 process where the primary acceptor of carbon dioxide, that is RUBP, is accepting carbon in the form of carbon dioxide and resulting in the formation of first stable compound called as phosphoglyceric acid. Otherwise we can call it as 3-phosphoglyceric acid or glycerate also. So as this is an 3-carbon compound, that's the reason why we call these plants as a C3 plants. And related to the question, the immediate product of carboxylation in C3 plants is phosphoglyceric acid. So let us discuss about the other options that have been given there. That in the first option it is given as RUBP. RUBP is a primary acceptor of carbon dioxide in C3 plants, whereas OAA, oxaloacetic acid, we call it as. So oxaloacetic acid is the first product or first stable product formed in C4 plants. So here there is a small variation that we can discuss here itself, that is C3 plants and C4 plants we are saying. Wherever the plants in which the first stable compound is an 3-phosphoglyceric acid, which is a 3-carbon compound, such type of plants are called as C3 plants. The plants in which the first stable compound is a oxaloacetic acid, that is OAA, which is a 4-carbon compound, that's why we call that as those plants as C4 plants. And GAP. GAP is one of the intermediate compound formed in the process of carbon dioxide fixation. So from the given options, the first or immediate product of carboxylation C3 plants is phosphoglyceric acid. So the correct option for question number 1 is option B. Question number 2. Which of the following influences feedback inhibition of photosynthesis? The given options are A. Chlorophyll, B. High light intensity, C. Low carbon dioxide concentration, D. Carbohydrate accumulation. So this question is related to the feedback inhibition. Actually, this is a concept related to the enzyme action. So in the enzyme action, we know that 3 concepts, 3 inhibitors are there. Those are called as competitive inhibitors, non-competitive inhibitors, and third one is feedback inhibitors, or the process is called as feedback inhibition. So why does this feedback inhibition occurs in plants? Means it is because of the accumulation of the end product. So here, how does this accumulation of end product will show effect on the process? Means here whatever the end product which is formed as a result of a chemical reaction, that end product will show its effect on the enzyme which is participating in the first reaction. So here, as we know that in the process, glucose plus ATP give rise to glucose 6-phosphate. Here the enzyme which is taking part here, it is a hexokinase enzyme. So this is a product formed as a result of this reaction, and this end product will show effect on the enzyme which is taking part in the first reaction. That is because of the accumulation of this end product. So it is not or we can say it is inhibiting the activity of the enzyme. When the activity of this hexokinase have been inhibited, so that will not take part in this reaction. When the hexokinase does not take part in this reaction, the product will not be formed. That is what we can say that accumulation of end product is the reason for feedback inhibition. So from the given options, option D is the correct answer. Question number 3. What type of reaction occurs when malic acid is converted into pyruvic acid in the bundle sheath cells of C4 plants? The given options are A. Decarboxylation, B. Dehydrogenation, C. Oxidative decarboxylation, and D. Transamination. So this is a question related to the C4 plants. In C4 plants, a special feature is there, that is the presence of Kranz anatomy, and also chloroplast dimorphism is present. Apart from that one, the reactions will show spatial differentiation. That is, some of the reactions will occur in the mesophyll cells and followed by the reactions will occur in the bundle sheath cells. In the mesophyll cells, the carboxylation will take place, that is the carbon dioxide accepted by the primary acceptor of carbon dioxide, that is PEP. So which will be converted into oxaloacetic acid. That oxaloacetic acid will be converted into malic acid in the mesophyll cells, and that malic acid will be transmitted from mesophyll cell to the bundle sheath cells. Once this malic acid, which is an 4-carbon compound, enters into bundle sheath cells, it will undergo a process called as losing of carbon dioxide, that is what we call it as a decarboxylation. At the same time, here oxidation is also been taking place, that is by so here in this process what happens is oxidation by loss of hydrogen, that is what we call it as dehydrogenation process. So the two steps are taking place where NADP is accepting a hydrogen which is lost from malic acid and converted into NADPH. At the same time, the malic acid will be losing carbon in the form of carbon dioxide, and it will be converted into a 3-carbon compound called as pyruvic acid. So here this is a step which is taking place in the bundle sheath cells of C4 plants. So here what is the actual step means, here apart from the oxidation process, that is dehydrogenation, decarboxylation is also taking place. That's the reason why we can say that the two steps, that is oxidation and decarboxylation, or we can call it as oxidative decarboxylation, will be taking place in the bundle sheath cells when malic acid is converted into pyruvic acid. So from the given options, option C is the correct answer. Question number 4. Which of the following isomeric reaction occurs in regeneration phase? 1. Ribose 5-phosphate giving rise to ribulose 5-phosphate. 2. Xylose 5-phosphate giving rise to ribose 5-phosphate. 3. GAP is giving rise to DHAP, and 4. DHAP giving rise to GAP. The given options are A 1 and 2, B 2 and 3, C 3 and 4, D 2 and 4. So here this is a question related to the C3 process or C3 pathway, in which actually C3 cycle we can call it as. In C3 cycle 3 steps are there. So first one is called as carboxylation phase, where in the initial question we had discussed that carbon dioxide accepted by RUBP, that is called as carboxylation, and the second step is called as reduction process, and third one is called as a regeneration. So now the question is related to the third step, that is a regeneration phase. So here in the regeneration phase some isomeric reactions are taking place, that is the two compounds which are having the similar molecular formula are converted here, that is one type of isomeric form is converted into other type of isomeric form. So here whatever the options that have been given, the 4 options questions, the 4 options that have been given the question, all of them are the isomeric reactions only. But the thing is whether out of those 4, how many are occurring in the regeneration phase, that is in the third phase of C3 pathway. So let us take first one, ribose 5-phosphate is giving rise to ribulose 5-phosphate. The second one is xylulose 5-phosphate giving rise to ribose 5-phosphate. And the third one is GAP is giving rise to DHAP, and the fourth one, the DHAP is giving rise to GAP. So out of these 4, as I said that all the 4 are isomeric reactions only, but the thing is whether out of these 4 how many are taking part in the regeneration phase of C3 plants. Here these 2 are the steps which are taking place in the regeneration phase, but these 2 are not taking part in the regeneration phase of the C3 pathway. So from the given options, 1 and 2 are the correct options. That is option A is the correct answer. Question number 5. Which of the following statement is not correct with reference to C4 plants? Options are A. Cran's anatomy, B. Dimorphic nature of chloroplasts, C. Agranule chloroplast in bundle sheath cells, and D. Chloroplasts of mesophyll cells store starch. So here this is a question related to C4 pathway or C4 plants. So as we had already discussed that C4 plants are those in which the first stable compound is an 4-carbon compound, that is oxaloacetic acid. So here which of the following is not correct, or we can say we have to identify the false statement related to C4 plants or C4 pathway. Let us take the first one, that is A. Cran's anatomy. So this is a special anatomical feature that is seen in the C4 plants, where around the we can say that around that xylem and phloem, that is a vascular tissue, the bundle sheath cells are present. The bundle sheath cells are in the form of a wreath-like arrangement. So the wreath-like arrangement of a bundle sheath is nothing but the Cran's anatomy. So this characteristic feature is present in C4 plants. Then B. The dimorphic nature of chloroplasts. So here in C4 plants 2 types of cells are participating. So those are mesophyll cells and other is a bundle sheath cells. Here dimorphic nature of chloroplast means out of the 2 types of cells, that is mesophyll and the bundle sheath cells, one type of cells will have granule chloroplast. So the meaning of the granule chloroplast is which contains both grana and stroma are present. While in some cells we call it as agranule chloroplast. So the meaning of agranule chloroplast means the cells which are having the chloroplast in which grana is absent in the chloroplast of those cells. That means only stroma is present. So this type of deviation can be seen. That's where dimorphic nature of chloroplast is present. This is a true statement related to C4 plants. Then agranule chloroplast in bundle sheath cells. Agranule chloroplast in bundle sheath cells. So here as I said that some of the cells will have granule chloroplast, some of the cells are having agranule chloroplast. Agranule chloroplasts are present in bundle sheath cells and the granule chloroplasts are present in mesophyll cells. So this statement is also correct related to C4 plants. Then coming to chloroplasts of mesophyll cells store starch. Chloroplasts of mesophyll cells store starch. Actually the steps will start in the mesophyll cells in the C4 plants, that is carboxylation will be taking place where oxaloacetic acid is formed, that will be converted into malic acid, and that malic acid Like if you consider, this is a mesophyll cells and these are the bundle sheath cells. So this is a step, this is a cell where the carbon dioxide is accepted in the form of bicarbonates. That the primary acceptor is the phosphoenol pyruvic acid that will be converted into oxaloacetic acid, and oxaloacetic acid is converted into malic acid. This malic acid will be transmitted from mesophyll cell to the bundle sheath cells, where the malic acid is undergoing oxidative decarboxylation. That is, carbon dioxide is lost and also hydrogen will be lost and converted into pyruvic acid. This pyruvic acid will enters into mesophyll cells and it will be once again converted into phosphoenol pyruvic acid. In this step, where the carbon decarboxylation have been taken place, where here RuBP will be present in the bundle sheath cells, which will be accepting this carbon dioxide and helping in the formation of starch. That means in C4 plant, the starch is formed in the bundle sheath cells but not in the mesophyll cells. But in the option, they had given that chloroplasts of mesophyll cells store starch. So it is not in the chloroplasts of mesophyll cells, but it is in the bundle sheath cells. So from the given option, the false statement or the wrong statement related to the C4 plants is the fourth one. So the correct option is option D. Question number 6. C4 plants are different from C3 plants with reference to options: A, the substrate that accepts carbon dioxide in carbon assimilation; B, type of end products of photosynthesis; C, number of ATP consumed in the synthesis of sugar; and D, the type of pigments involved in photosynthesis. So here this is a question related to comparison between C4 and C3 plants or C3 mechanism. Let us come to the first one, the substrate that accepts carbon dioxide in carbon assimilation. So here in C4 plants, the primary acceptor of carbon dioxide is phosphoenol pyruvic acid, that is PEP, whereas in the C3 plants it is RuBP. That is, the primary acceptor of carbon dioxide during the assimilation process is different in both the plants. The substrate that accepts carbon dioxide in carbon assimilation is different in both the plants. And let us come to option B, that is the type of end products formed in both the plants. So actually, irrespective of whether the first compound, the nature of the first compound or the nature of the primary acceptor, the final product in both of them will be starch only. So there is no differentiation in the end product of the photosynthesis. And of course, when it comes to C, the number of ATP that have been consumed for the synthesis of sugar will be same for both of them. And the type of pigments which are involved in that one. So here, as we know that the pigments which are involved are the chlorophyll pigments only, along with the accessory pigments, that is carotenoids. So there is no nature of there is no difference in the nature of pigments which are involved in photosynthesis of C3 plants and the C4 plants. So from the given options, we can say that the substrate which are involved in accepting the carbon dioxide are different in both the plants. So from the given options, option A is the correct answer. Question number 7. In photosynthetic quinone cycle, the number of electrons transferred from PQH2 to cytochrome B6 is. The given options are A 1, B 2, C 3 and D 4. So this is a question related to the electron transport during photosynthetic process. So actually, in the photosynthesis, during light cycle or during light reaction, the electrons are actually, to say, the photolysis of water will be taking place. That is, the water will split to produce oxygen, electrons and protons. These electrons are accepted by the photosystem 2. Those before that, what happens, the photosystem 2, the reaction center of photosystem 2 will lose electrons by excitation. So the loss of electrons in PS2 are compensated by photolysis of water, that is the 2 electrons that have been released here. So these electrons that have been lost from PS2 will be carried through different electron carriers like pheophytin and some other compounds, cytochromes and also plastoquinone, all these things. So where the quinone cycle will be operated. Quinone cycle will be operated. So where semi-plastoquinol is accepting that electrons and also the protons from the other side of the membrane of chloroplast and get reduced, converted into PQH2. And from here the electrons which have been taken from here will be given to cytochrome B6. So here whenever the quinone cycle is operated, only one electron is taken here. So 2 protons and 1 electron is taken. So whatever the electron that have been taken by this completely reduced plastoquinone, so that will be transmitted to cytochrome B6. In the meantime, the protons will be released into the other side of this membrane. So that means only 1 electron is transmitted from PQH2 to cytochrome B6. So that is the way how in the electron transport system, every time we consider 1 electron will be transmitted from 1 electron carrier to the other electron carrier. So from the given options, option A is the correct answer. Question number 8. Red drop is due to the given options: A, decline in quantum yield in separate beams of red light; B, non-functioning of PS2, that is photosystem 2 in far red light; C, inhibitory effect of red light on photosystem 1, that is PS1; and D, oxidation of chlorophyll. So here red drop. So this is a concept where the quantum yield will decrease. So here the thing is actually the higher wavelength of red light. Whenever the higher wavelength of red light is taken, there is a drop in the decline in the quantum yield of the photosynthetic rate will be declined or decreased. So here what is the reason for decline in the photosynthetic rate means it is because of the non-functioning of photosystem 2. That is because of the non-functioning of photosystem 2, that is PS2. So this is the wavelength of far red light. So this is a main reason for red drop. So from the given options, option B is the correct answer. Question number 9. Photosynthesis. The given options: A, physico-biochemical process; B, anabolic process; C, endergonic reaction; and D, all the above. So here this is the basic question related to photosynthesis. So we always say that a photosynthesis will take place in plants, but basically what is photosynthesis? So actually here, as we know that this is one of the physiological process or metabolic function that is taking place in the plants. That's why we call it's an physiological process, or we can call it as metabolic process, where some compounds are converted into other type of compounds. So here, as it is taking place inside the living organism, so we call it as bio process. And here it is the chemical process, we can also call it as. Why we are calling it as a chemical process means, here inside the cell, inside the living cell, one type of compound is converted into other type of compound. That's the reason why we can say the chemical reactions are taking place. That's why we call it as a biochemical process. And B, it is an anabolic process. So here in the mechanisms, we know that anabolic process and catabolic process are there. Anabolic process or anabolism means it's a constructive process where simpler compounds are converted into the complex compounds. That's what we call it as anabolic process. Why photosynthesis is called as anabolic process means here the simpler compounds like carbon dioxide and water will be converted into the complex compounds like one of the example is glucose molecule. So this is a basic thing that is happening, so we call it as a anabolic process. And C, that is endergonic process. So why we are calling it as endergonic process means here this is a step where energy is utilized. So why the energy is utilized means because to convert a simpler compound into a complex compound, there the energy is utilized to form the new bonds between the simpler compounds to produce a complex compound. That's the reason why energy is utilized, and as the energy is utilized we call it as a endergonic process. So from the given options, all the three are related to photosynthesis. So from the fourth option, all the above options are correct. So that's what we can say that as A, B, C are related to photosynthetic process. So we can say all the three options that is A, B, C are correct. That means option D from the given options is the correct answer for this question. Question number 10. The overall reaction of the Calvin cycle is. So here this question is related to the overall reaction, that is given options. Option A, 3 RUBP plus 3 carbon dioxide plus 9 ATP plus 6 NADPH plus H plus giving rise to 3 PGA plus 9 NADP plus 9 inorganic phosphate plus 6 NADP plus. Option B, 3 RUBP plus 3 carbon dioxide plus 6 ATP plus 6 NADPH plus H plus giving rise to 3 PGA plus 6 ADP plus 6 inorganic phosphate plus 6 NADP plus. Option C, 3 RUBP plus 3 carbon dioxide plus 9 ATP plus 6 NADPH plus H plus giving rise to 6 G3P plus 9 NADP plus 6 NADP plus 9 inorganic phosphates. Option D, 3 RUBP plus 3 carbon dioxide plus 6 ATP plus 6 NADPH plus H plus giving rise to 6 G3P plus 6 ADP plus 6 NADP plus 5 inorganic phosphates. So here this is related to Calvin cycle, that is a C3 cycle, where the 3 RUBP molecules when they are participating, so one RUBP molecule will accept one carbon dioxide molecule. That means in all the given options it is taken as 3 RUBP molecules only. That means they will accept 3 carbon dioxide molecules. That means each RUBP will accept one carbon dioxide, and for this process they utilize the energy in the form of ATP. So ATP will be utilized here when one carbon dioxide molecule is accepted by one RUBP molecule, they require 3 ATP molecules. So as it is 3 carbon dioxides, so we can say that the number of ATP that are required will be 9 ATP molecules. And here apart from this one, the NADPH plus H plus are also being used. So here for one carboxylation or when one carbon dioxide is accepted by one RUBP molecule, 2 NADPH are utilized. So that means for 3 compounds, 6 NADPH plus H plus are utilized. So these are the compounds related towards the substrate side, and they will give rise to 6 molecules of G3P. That means we can say glyceraldehyde 3-phosphate. So G3P pool, a total of 6 G3P molecules will be formed, and by using 9 ATPs. So 9 ATPs, they are losing 1 phosphate each and converted into 9 ADP molecules and 9 inorganic phosphate molecules, and 6 NADPH are converted into 6 NADP molecules. So this is the overall reaction where 3 molecules of RUBP, 3 molecules of carbon dioxide, 9 molecules of ATP, 6 molecules of NADP H plus H plus are giving rise to G3P pool, which contains 6 molecules, that is 6 G3P molecules, 9 ADP plus 9 inorganic phosphate molecules, and 6 NADP molecules are produced. So from the given options, option C is the correct answer for this question. Question number 11. Choose wrong combination with respect to the location of enzymes in C4 plants. The given options are PEP case stroma of bundle sheath cell chloroplast, B. Rubisco stroma of bundle sheath cell chloroplast, C. Malic enzyme stroma of bundle sheath cell chloroplast, D. Pyruvate dikinase stroma of mesophyll cell chloroplast. So this is a question once again related to the C4 plants. So as we have discussed that in C4 plants, the spatial differentiation is taking place during the photosynthesis, where it is taking place in 2 types of cells. One is called as a mesophyll cell and other is called as a bundle sheath cells. So the process will start in the mesophyll cells and continued into the bundle sheath cells. Here the primary acceptor of carbon dioxide is the phosphoenol pyruvate, that is a 3 carbon compound, that is PEP, and that will be converted into a 4 carbon compound called as oxaloacetic acid, which is a first stable compound in C4 plants, and this compound is converted into malic acid, and the so formed malic acid will be transferred from mesophyll cell into the bundle sheath cell, where it will undergo oxidative decarboxylation by losing carbon dioxide and also by hydrogen, where it will be converted into pyruvic acid, and this pyruvic acid once again will be entering into the mesophyll cell, where it will be converted into phosphoenol pyruvate. Here the carbon dioxide is accepted by RUBP, which is present in the bundle sheath cells in presence of an enzyme called as Rubisco enzyme, and converted into or helping in the formation of starch molecule. So here this is a brief substances which are formed during the C4 cycle, and here for the first step the PEP case is an enzyme which is required where carbon dioxide is accepted by PEP to convert into oxaloacetic acid, and this is the first one, that is PEP case, which is occurring in the mesophyll cell chloroplast, and malic enzyme is required here, which is in the bundle sheath cells. And Rubisco enzyme is also been present in the bundle sheath cells only. And pyruvate dikinase is utilized in this step, where pyruvic acid converted into phosphoenol pyruvic acid. So pyruvate dikinase in the mesophyll cells, chloroplast. So these are the 4 enzymes that have been taken in the question. So here from the options here they are given that PEPcase is present in the chloroplast of bundle sheath cells is a wrong option, whereas the other 3, that is malic enzyme present in the chloroplast of bundle sheath cell, rubisco is present in the chloroplast of bundle sheath cell, pyruvate dikinase is present in the mesophyll of the chloroplast of mesophyll cells. So from the given options, option A is the correct answer. Question number 12. Number of triose molecules from G3P pool that enter into cytosol from chloroplast to form the main end product of C3 cycle, which is transporters through phloem. The given options are A 2, B 10, C 4 and D 6. So here G3P pool. So here in the C3 plants, so here the G3P pool is formed in the reduction phase, that is the second step, where some of them are helping in the regeneration of the process, that is whatever the number, that is 5/6 of G3P are used in regeneration of RUBP, while 1/6 of G3P present in G3P pool are used in the production of the starch or the formation of the end products which will be transported through the phloem tissue. So here the number of G3P that have been formed are actually when 3 carbon dioxide molecules are taken. So that is 3 carbon dioxide plus 3 RUBP. It will be resulting into 6 molecules of G3P. 6 molecules of G3P. So here as 5/6 means that is out of 6, 5 will be used in the regeneration of RUBP, whereas 1/6 means only one of the G3P is used in formation of the photosynthesis, which will be transported through the phloem tissue. But as we know that 1/5 means only one G3P molecule, so which is a 3 carbon compound. So whereas the end product is minimum, it should be a glucose molecule. To produce glucose is a 6 carbon molecule. That means instead of 1 G3P, they require 2 G3Ps to produce 1 glucose molecule. But in the question, that is a starch molecule. So the starch molecule requires a total of 4 G3P molecules. That means here instead of 3 carbon dioxide molecules, the number should be 12 carbon dioxides plus 12 RUBP, giving rise to the number that is related to that one, 12 G3P molecules. So 24 G3P molecules. So when 24 G3P molecules are produced, that means 5/6 are used in regeneration of RUBP, that is 20 G3Ps, whereas the 1/6 of 24, that is 4 G3P are used in the production of the photosynthesis. So from the given option C is the correct answer.